So we made our dehydrogenase buddies, and now we're going to move those electrons around in what we call the electron transport chain. So big picture first, in your mitochondria, you have these series of complexes. That, with the help of our dehydrogenase buddies, move electrons around. And that creates a current, kind of like when you plug in your electronics, there's a current that powers something. Well, here there's a current that powers something, and that powers the release of H+. You're going to release a lot of hydrogen up, and what goes up comes down. Comes down through this ATP synthase. And when it does that, it generates ATP. And that is the big picture. Now let's get into the nitty-gritty facts. First off, you need to know what these complexes are. So, complex 1 is also known as flavin mononucleotide, or FMN. Complex 2, here's a big hint, FADH2. What made FADH2? Succinate dehydrogenase, right? And didn't I say that was part of the ETC cycle? Well, here it is, clear as day. There's also a friend that stuck on there called Coenzyme Q. And the thing you need to know about this is that this is the only one that isn't a protein. It's actually from lipid sources. All right, complex 3 is going to be your cytochrome BC. And then complex 4, the thing that helps move electrons in complex 4 is going to be oxygen. That's why oxygen is so important in generating ATP. And because it uses oxygen, we call this complex cytochrome oxidase. And then finally, this is the ATP synthase I was talking about. Sometimes split up into F1 and F0. F1 is just the head that sticks out. F0 is more or less the body. And that is it. One thing to note, FADH2 skips a step, starts with one a little earlier. So FADH2 actually makes less ATP than its friend, NADH2. Let's see if I missed anything. I think that does it just for a general overview and the small facts that you do need to know. This is how it usually works. Now, as clinicians, we're probably not worried so much as how it usually works, as in how it can go wrong. So let's talk about how it can go wrong. So there are a few things that can go wrong with this electron transport chain. First one we're going to talk about is hypoxia. Second one is mitochondrial disorders. And then uncouplers. And lastly is inhibitors. So we'll start with hypoxia. In hypoxia, you don't have O2, and if you don't have O2, can't finish your last step, don't have ATP, and that's how you die from hypoxia. Done. 2, mitochondrial disorders. There are 3, and the name gives it away. So you have Leber's, optic neuropathy. There's a defect in complex 1 and causes blindness. 2 is MELAS, M-E-L-A-S. This is short for mitochondrial encephalomyopathy. You can tell that just by the name gives it away. You're going to have lactic acidosis, muscle weakness, and you're going to have encephalopathy and strokes. So strokes and lactic acidosis is key. And this is due to a leucine mutation in your tRNA. Lastly is going to be Murf. This is myoclonic epilepsy with ragged red fibers. That's what Murf stands for. And again, the name gives it away. You're gonna have seizures. That's your myoclonic epilepsy part. And then without ATP, your muscles are gonna die and they're gonna look ragged under microscopy. And they'll tell you that usually in the question stem. Usually I've seen questions say, you know, a patient has epilepsy, seizures, muscle weakness, they do a biopsy and they see ragged red fibers or just ragged muscle fibers in general. And then they'll give you a pedigree and say, what does it look like on a pedigree? Well, these are mitochondrial disorders, right? How do mitochondrial disorders look on a pedigree? The mother's children all have it, 100%. That's how it works. We'll talk about pedigrees in a later video, but that's usually how they ask the questions. Just know those. Done with mitochondrial disorders. Uncouplers. What the heck does uncoupling mean? This is what it means. H plus falling down in our ATP synthase is a match made in heaven. That's what we want. That's what we're trying to do. This is the perfect couple. Uncouplers make sure H plus falls outside of it. Now that's not good, because we generate all this current, all this power to power H plus, hoping that it goes into our ATP synthase, gives us ATP. Uncouplers, we generate all this power, pump out H plus, and it doesn't fall through ATP synthase. That uses a lot of power, that uses a lot of energy, generates a lot of heat. So what you see in uncouplers is that you see hyperthermia. Now, there are a couple uncouplers. Let me just clear the board. One of them is physiological. Babies have something called thermogenin, or brown fat. That's what keeps them warm. It keeps them warm because it uncouples this, powers this, H plus doesn't come through, so it generates a lot of heat. Keeps babies warm. Another thing is 2,4-dinitro phenol, which is a pesticide. Same thing. And you're telling me, well, that's not really, you know, one is physiological, one we don't really see. Is there something more common? And yes, the most common is aspirin. Don't aspirin overdoses cause hyperthermia? Ever wonder why that was? It causes uncoupling. That's what causes the hyperthermia. That does it for uncoupling. Lastly is going to be your inhibitors. What does inhibitors mean? Inhibitors mean that they basically inhibit a complex. By inhibiting a complex, you don't have the current, you don't have the ATP. We'll start with complex 1. Barbiturates inhibit complex 1. Rotenone has 1 in his name. That's another pesticide. Inhibits complex 1. And then MPTP. You may have remembered that from your neuroblog. MPTP is that synthetic drug that caused acute Parkinson's. Yeah? Please know that one. Complex 2. That's that pseudo-complex, the one that's from a lipid source. Yeah? Complex 2. Another pesticide, carboxin. Pesticides really are no good. And then lastly, you need to know that Coenzyme Q, its little partner, is a ubiquinone. And that is blocked by atavaquone, which is an anti-malarial, but this anti-malarial is a ubiquinone analog. It's also blocked by statins, because statins work on your lipid synthesis decreases coenzyme Q synthesis. And one of the main side effects of statins is rhabdomyolysis. Everyone know why that was? Because it basically stops ATP in your muscles, causes rhabdomyolysis. Complex 3, antimyosin with complex 4. This is the things that work on your respiratory system. Remember, this is cytochrome oxidase. This is what uses oxygen. So, CO binds heme, stops oxygen from binding. Cyanide, same deal. And then finally, we have our ATP synthase. The inhibitor of ATP synthase is oligomycin. This is an antibiotic, too toxic to use, and the reason it's too toxic to use is because it blocks ATP synthase. I think that does it. Again, know just kind of generally what goes on. Know why hypoxia causes problems, the mitochondrial disorders, uncouplers, the mechanism behind that, and then finally the inhibitors. All the inhibitors. And I think you should be good. That does it for this video. See you next time.